The overall goal of this framework is to identify genes involved in broad-range dietary restriction, quantify the environmental information encoded by their expression levels, and understand the underlying coding strategy that links environment to lifespan.
This method can help answer key questions in the neurobiology of aging field, such as which genes convey information from the environment to modulate lifespan.
Although this framework can provide insight into the C. elegans sensory system, it can also be applied more generally to any biological system whose components cooperate to process environmental information. The main advantage of this technique is that it quantifies the environmental information encoded by groups of neurons and determines the coding strategy employed by neural circuits to convey this information to downstream targets.
After culturing OP50 on NGM plates according to the text protocol, prepare 500 milliliters of LB medium in each of 2-liter Erlenmeyer flasks and autoclave them. Inoculate each flask with a single colony of OP50 and grow the cultures at 37 degrees Celsius and 200 RPM for around 14 hours. Next, supplement the cultures with 50 micrograms per milliliter of streptomycin. Then shake the flasks for an additional 30 minutes at 37 degrees Celsius before placing the flasks on ice for 15 minutes. Transfer 450 milliliters of each culture into separate 500 milliliter sterile centrifuge bottles. Retain the leftover culture on ice as it will be used to determine the concentration of the bacteria. Spin down the bottles at 4500 times G and 4 degrees Celsius for 25 minutes. Then discard the supernatant and store the bottles on ice.